very much. And first, I'd like to say that what I'm speaking about today is really not in my area of expertise, but it's something that we're very excited about. And we got working on this because of really wonderful collaborations, as you can see, around the university. Specifically, it's a collaboration with physicians at the hospital, Richard Clark and Dennis Galanakis, and Professor Marcia Simon from Oral Biology and Pathology in the School of Dental Medicine. What raised it, it just kept growing, and then it raised it to the next notch through our collaboration with Yufeng Deng and Peng Zhang from the Department of Applied Mathematics and Statistics that added a modeling component. And throughout all this, I mean, we knew nothing about virology. Was really tremendous help from Stefan Müller, ex from Stony Brook as well, and Fubao Lin, who works with Dr. Clark there, and they have formed companies. The work that I'm showing here is really, majority of it's a PhD thesis of Kao Li, and together with assistance from Quanche Feng, who's now our resident virologist. And the theory component comes from Bernard Essuman, and we have, in addition, participation from our summer program, Ikshu Pandey from Johns Hopkins, who's here today. And as you can see down here, we're very proud to have participation of a whole host of high school students when this work was also in full blown this summer. So it's an organic piece of work that has grown. And we really, the reason I'm presenting here, as Nina said, is because we really would welcome collaboration and comments from people. And my internet is very slow, so I hope I may have to go to, okay. Infections, just to lay the groundwork here and to answer any pre-comments before they start, and an explanation of why we're doing what we're doing, is first of all, infections in general are thrombotic diseases. And I learned this just recently reading lots of review papers. And just about viral infections, Ebola, we know very well, but also influenza as well as COVID results in thrombosis. And what we have is just a quick comparison between the flu virus and SARS-CoV-2. This is a SARS-CoV-2 symposium. And the reason why we're not talking about SARS-CoV-2 directly is it requires BSL-3 access with instrumentation that is not available at Stony Brook right now, even if we were to go into the BSL-3 enclosure. And we were a little bit nervous about jumping in there. So the influenza virus, H1n1, a great deal is known about it already. And it as well is a thrombotic disease. A lot fewer cases right now, given what happened to SARS-CoV since this little blurb got written, and it's also less toxic, 0.096% morbidity. But in terms of the way it works and the physics of what it does in inflammation, there are many, many similarities. So we focus on the H1n1 virus. And in some sort of the big picture of what happens, and again, the basic differences between the two, because this is especially important when people start to simulate and to calculate. The H1n1 virus infects by binding of hemagglutinin, which is expressed on the surface of the virion, and they look for sialic receptors on the cell membrane. It fuses with the cell membrane, and then gets absorbed, releases its RNA, and a huge cascade of different things begin to happen. Similarly, SARS-CoV-2 infects the epithelial cells, and I should say that H1n1 does the same. It's the epithelium. Eric Mackow at Stony Brook has shown this very well, that endothelial cells do not get infected. But the epithelial cells emit all kinds of cytokines, chemokines that then do make their way and have a lot of sequelae. A recent paper from Cell actually looked at which genes are upregulated and in a timeline for how long it takes to upregulate them. And this was infection of endothelial cells was PR8, it's the Puerto Rican wild type H1n1. And this line is drawn at 2 hours after infection. C2 are these interferon sensitive genes of the epithelium. And you can see that already upregulation, massive upregulation of these interferon cells, sensitive cells, already occur 2 hours after infection of MDCK epithelium cells. And one second. Again. Okay. So what happens after the infection? Okay. This is the endothelium. There's this massive cytokine storm that occurs in both of them. And these factors then go through and they permeate to the endothelium, causing all these are tons and tons of papers that have appeared recently about aberrant coagulation that occur both with SARS-CoV-2 and here the important part, other pandemic viruses. And the conclusion of all these papers is that even though we know that they hamper pathogenic, even though a lot is known about this in all these body of literature, a good model, a detailed mechanism of how the coagulation contributes to pathogenesis of even influenza, let alone SARS-CoV-2, which is more recent, has to be investigated at a molecular level. Now, what have they got in common? What you see here is a giant thrombi, which we got from Mohsen Banazadeh. He's a vascular surgeon at Stony Brook. And you can see how big they are. That is about 5 or 6 inches long in somebody's leg. This was a former SARS-CoV patient. And they come out in one piece. They're not fragmented. It looks like almost like when you stuff something and it just molds to the shape. What you're seeing in these other images right here, especially with the arrows that's microthrombi formation. These are in alveolar capillaries from SARS-CoV, and you get exactly the same thing, is also well known after H1 infection. And H1 infection in lungs of people who don't make it look very similar to the x-rays of people after SARS-CoV-2 because these microthrombi prevent the exchange of oxygen and blood levels go down. So the question is, why does that happen? And the normal thrombotic cascade requires thrombin. There's no thrombin here. These are the very initial parts of the thrombosis that then really results into something like this. Now, again, when we're thinking out of the box, when we saw a really nice talk by Richard Clark, and Richard and Fu Balin have a company that makes a peptide called P12. These are 12 amino acids from the fibronectin molecule. Fibronectin binds to fibrinogen, it will show. And what they found was the addition of P12. This is completely off field from here, but very related. The addition of P12 prevented burn progression from 2nd to 3rd degree burns. And the reason that they showed was that this is a capillary very similar to the ones you see up here. So the capillary on the bottom forms lots of red blood cell clots as soon as there is injury to the capillaries from the 2nd degree burn. In the absence of anything, microthrombi form, you get necrosis and the wound progresses. With the addition of P12, he prevents the microthrombi from forming. He doesn't prevent the fibrinogen, which is red and stained, but he prevents microthrombi formation. So the question is, what does P12 do to prevent these thrombotic events? How does it work, and why do thrombotic events occur altogether when a surface is damaged? So out-of-the-box hypothesis here is that there's a similarity between microthrombi formation with COVID or H1n1 and burn progression. How are they similar? Now, because I'm a physicist, a lot of these events, to a physicist, everything looks the same. You have a hammer, everything, the world's a nail. But sometimes it isn't. And sometimes we hypothesize that there are valid reasons for comparison. So just as a very quick review, this is a fibrinogen molecule. The fibrinogen molecule has alpha C domains that cover it, which are hydrophilic, and the interior is hydrophobic. So normally when you have a cascade in a wound, thrombin is released. Thrombin cleaves the fibrinopeptides. It releases the alpha C domain. They are alpha C binding domains. Keep that in mind. Very important. Then recruit other liberated fibrinogen molecules, and fibers begin to form. Problem: when you have the burn and when you have COVID, there is no thrombin released in the very, very first few minutes of this cascade. So the fibrinogen molecule is very sensitive to the structure of surfaces. And what you're seeing here, this is older data back from 2005, or was it ours, from the literature. If the surface becomes hydrophobic, the interior of the fibrinogen molecule adsorbs, and it adsorbs flat with enough energy to mimic thrombin so that the fibrinopeptides are released and the fibrinogen molecule is now open. On a hydrophilic surface, on the other hand, the alpha C domains, which are hydrophilic, are now attracted to the surface, and the fibrinogen molecule has a totally different conformation. What are the consequences here? And which are the segments that are necessary for forming fibers? Dr. Galanakis is really one of the treasures that we have at Stony Brook, and in his entire career has spent producing antibodies and knowing every single piece of the fibrinogen molecule. And by a selective binding to different parts of the fibrinogen molecule, we determined that the regions responsible for making the fibers are the very edges and the regions at the very edge of the alpha C domain. So that, sorry, this is gonna, okay. If you block the D domain, nothing adsorbs to the surface, the molecule is released. If you block the 518 to 584, the tips of the alpha C domain, not much will happen. Fibers won't form because you can't recruit any other chains. Whereas if the molecule is wide open, turned inside out, with the hydrophobic domains adhere to the surface, the alpha C domains are exposed, and other molecules can be recruited from the bloodstream. What are the other molecules? And this is from a recent paper that Dr. Galanakis just submitted. The other molecules are what's known as the soluble fibrin fraction of blood. Sorry. The soluble fibrin fractions of blood are small segments of fibrin that formed when there was some event, thrombotic event, such as you were hurt and not all the thrombin was removed. People who had electrophoresis, people who underwent dialysis, they all have a lot of soluble fibrin. And this is a SEM image of a blood clot from the Wiesel lab at UPenn, where you can actually see the soluble fibrin that sort of absorbed as blobs onto the other fibrin fibers. The soluble fibrin has fibrin on the outside, 3 alpha C domains, and they are easily recruited on hydrophobic surfaces, which you see over here, where a hydrophobic surface is defined as any surface, polymer, any surface that has a contact angle which is greater than 60 degrees. Surfaces such as P4VP, silicon wafer, surfaces that have been made hydrophilic by UV ozone plasma, for example, where the contact angle is below 60 degrees, do not form spontaneously these massive fibrinogen fibers. It's kind of like a tree. They're anchored to the surface. And this is from data that we had done with Kat Vorvolakos at the FDA, where we're trying to actually develop a non-thrombotic surface. Here you have PS, PMMA, PLA. You can see these fibers by eye. You can see the large bar here. P4VP, no fibers. Here are fibers from the way that polymer is structured. BCA analysis showed that you have plenty of fibrinogen adsorbed even to the PVP surface. Just the fibers cannot initiate unless those domains are free. And here you have, if you have antibodies against those domains, you see that on the P4VP, lots of domains on the first layer. On the PS, you see them on the fibers because the fibers have the alpha C domains are free. On the P4VP, the alpha C domains right here are blocked because of the way the molecule is sitting, but the E domain is exposed, which is what you see up here. So using selective antibodies that we got from Dr. Galanakis, we know exactly how fibrinogen sits when it's exposed to different surfaces. Now, in addition to exposing the alpha C domain, they also expose domains on the gamma chain. And these domains on the gamma chains are specific to attracting platelets or red blood cells. And plenty of them are exposed also on the hydrophilic P4VP, which means that if you make a stent hydrophobic or hydrophilic, it's going to attract blood clots. And this, you can see, this was done again at the FDA where we were flowing a platelet-rich plasma through an artificial artery made by these polymers, and you can see a very nice blood clot forming on polystyrene with activated platelets. Similarly on PMMA with activated platelets. And on P4VP, you've got red blood cells coming up for a very nice red blood cell clot. You can see them right here. These are the clots, and all the domains are ripe for clotting on all surfaces. This is a calculation that Bernard Esserman did. Bernard, working with Peng Zhang and Yu Fendang's group, said, wait a minute, let me see what I can do. And he took the P12 peptide, which they know very well what it is from the fibronectin, and knowing that there's a fibronectin binding domain on fibrinogen, he started to play around and he said, where would this P12 bind on fibrinogen? And he's found, lo and behold, these purple things are P12 molecules right here. This is fibrinogen, this is P12, and he found that P12 binds on fibrinogen right here, right in the middle of the alpha C binding domain. That's what P12 looks like. And when you have it, it goes right in the middle of that alpha C binding domain. And the consequence is, this is optical microscopy, that's these beautiful fibers that you see coming up on polystyrene, which is hydrophobic. In this case, they added 50 micromolar P12, no fibers whatsoever, completely suppressed. Very similar when we put the antibody in that domain and no fibers occurred. What is a sequelae when we float platelet-rich plasma across it? Without P12, tons of fibers and initiation of clots. With 10 micromolar P12 or 100 micromolar, it didn't make that much difference. No fibers formed and none of the initiation of early clotting. Sort of in, this is flowing again, and this very similar to explain what he had observed, where even though, and this is again, amount of fibrinogen collected, plenty of fibrinogen collected, even with P12 that you see right here, but no fibers, because the fibers are what initiates non-thrombogenic clots. So how do we get between that and infection of H1N1? So we tried, these crazy experiments. Let's try something. And we plated MDCK cells. MDCK cells are dog epithelial cells, dog kidney epithelial cells. Or from Marcia Simon's lab, we had human oral epithelium. And they were infected with virus, an MOU, sort of a one virus particle per cell. And we followed standard protocol, infection for 1.5 hours in a rocker, and then 24 hours at 33 degrees. This is what you do for expanding the cells on MDCK. And then after all that, we washed away the virus and exposed it to fibrinogen. And you can see we have somewhat elevated fibrinogen, because that's exactly what happens in vivo when people are exposed to either H1N1 or SARS-CoV-2. Your fibrinogen levels go up almost by a factor of 2 in severe cases.
Miriam, we have only 4 minutes left in case you want to keep time for questions.
Okay, I'm going to go really fast because this is sort of the end. So we exposed it to fibrinogen, and what do we find? We find that here are the cells, and lo and behold, we see these huge fibers now form on the infected epithelium. And just like Dr. Galanakis had predicted, we only see it if there is soluble fibrin in the fibrinogen concentration. If you deplete the soluble fibrin, nothing happens. And if you add P12, nothing happens. And this is with direct infection. What happens with indirect infection? Again, remember what Professor Macau had said, that you don't affect, you're not infecting the endothelium, you're affecting the epithelium. So we did the same thing again. We took the media away after 1.5 hours and after 24 hours. And then we added the media to HUVEC cells. And we did this both from dog and human. And it's the same exact thing. Already after 1.5 hours, we didn't believe it. In the beginning, people said, no, that's ridiculous. We did it over and over again. After 1.5 hours, with these giant fibers, these fibers are almost a micron in length, almost a millimeter, in length. We see the giant fibers forming, but now they're forming on the HUVEC cells, on the endothelium. But P12 stops them. So, in conclusion, we think we've shown that H1N1 virus initiates non-thrombogenic fibers. Exposure to conditioned media on endothelial cells also generates the fibers. And P12, which is a therapy for burn wounds, may also provide a therapy against thrombosis and COVID. Thank you.